The overall goal of the following experiment is to use high-throughput sequential ELISA in order to efficiently analyze large numbers of novel biomarkers that were discovered by proteomics. This is achieved by first adding samples to a source plate, which is used to create dilutions for each biomarker and to store recovered plasma between tests. Next, ELISAs are run for each protein in order to determine their concentrations in patient samples. Ultimately, the elevation of the protein levels of these biomarkers at the onset of clinical manifestations of acute graft-versus-host disease can be assessed.
The main advantage of this technique over existing methods is that sequential ELISAs can analyze multiple novel proteins that are not always available with other multiplexing platforms.
The implications of this technique extend toward the diagnosis of acute graft-versus-host disease, as biomarkers validated through this method are now being investigated for clinical care.
Though this method can provide insight into complications in post-hematopoietic stem cell transplantation, it can also be applied to biomarker discovery and validation for other diseases, such as lung transplantation complications, lung cancer, or potentially any disease for which a diagnostic blood test will be useful.
Visual demonstration of this method is critical, as it is important that the multiple steps are done consistently to ensure reproducibility and low batch variability.
On the day before the experiment, spin-down thawed plasma aliquot samples for 10 minutes at 15,300 RCF and 4 degrees Celsius to separate the clots at the bottom and the lipids on the top from the plasma. Then, plate 150 microliters of undiluted plasma from each sample onto a 96-well V-bottom plate by manual pipetting according to predefined maps.